Hello children, Shubha teacher here. I am back to you with the fourth video of chapter photosynthesis. Till now, you have learnt about the entire process of photosynthesis, end results of the products of photosynthesis, and adaptations in leaves which facilitate the process of photosynthesis. We move on to understand the various factors which affect the rate of photosynthesis. There are four external factors and some internal factors. We start with external factors. Light intensity and carbon dioxide concentration. With increase in light intensity, the rate of photosynthesis increases. But we know that very high light affects the cell and also the enzymes. So therefore, to a certain limit, light intensity increases, increasing the rate of photosynthesis. Same goes with carbon dioxide concentration. Normal concentration of carbon dioxide in the atmosphere is 0.01 percentage. But with increase in carbon dioxide concentration in air, rate of photosynthesis also increases. But again, it is a poisonous gas. So it increases to a certain limit, helping increase in rate of photosynthesis. But maximum it is seen that at the concentration of 0.5 percentage of CO2 in air facilitates maximum amount of photosynthesis. Higher than that leads to closure of stomata. So therefore, photosynthesis is affected by these two factors in this way. Let us understand the graph shown here. Here this graph is having light intensity depicted in the x-axis and rate of photosynthesis being shown in the y-axis. Now you see the first line of graph. What is the light intensity or temperature shown here? 20 degrees centigrade with carbon dioxide percentage 0.1%. So at this temperature, the rate of photosynthesis increases and again becomes stable. When temperature is increased from 20 to 30 degrees centigrade, the rate increases again with the same constant carbon dioxide concentration. But again, when the temperature is lowered but CO2 is increased, again it is seen that the rate of photosynthesis increases. Again, with increase in temperature and CO2 at 0.2, again there is an increase. So we conclude that rate of photosynthesis increases with the increase in light intensity as well as with increase in CO2 concentration in the air. But there is a limitation to both. Temperature, excess temperature or very high light intensity affects. So to a certain extent, the rate increases but then becomes constant. And carbon dioxide concentration also from 0.1 to 0.5 percentage of CO2 content in the air increases the rate of photosynthesis. But after that, it lowers. We move to the next factor, temperature. The normal range of temperature at which living organisms function well, the body cells, enzymes, everything function well, is the optimum range 25 degrees centigrade to 35 degrees centigrade. Now again, temperature lower than 25 and higher than 35 degrees centigrade affects the rate of photosynthesis. It is seen a rise of 10 degrees C up to optimum temperature, that is within this range. If there is a jump of 10 degrees centigrade, rate of photosynthesis doubles. Like you have given some examples of temperature ranges. If the temperature rises from 20 degrees centigrade to 30 degrees centigrade suddenly, or 23 degrees centigrade to 33 degrees centigrade, all are within this range of temperature, then it is seen that the rate of photosynthesis doubles. The last external factor which affects photosynthesis process is water content. Now, since water maintains the normal concentration of the sap as well as the protoplasm inside the cells. If water becomes less or scarce in the stomata, it leads to closure of stomata. Because of water, there is turgidity in stomata, and because of that, stoma opens. We had discussed about the opening and closing of stomata, the mechanism of it, the potassium ion concentration theory, if you remember in the previous video. So, water content maintains the opening and closing of stomata. We come to the internal factors now. Chlorophyll. Since we know chlorophyll absorbs light and that initiates the process of photosynthesis. So if there is a loss of chlorophyll because of some nutrient deficiency, that lowers the rate of photosynthesis. Next is protoplasm. Protoplasm has got a normal chemical composition. If there is dehydration, that is, if there is a loss of water content, reduction in water content in protoplasm, that also reduces photosynthesis process. If suppose glucose which is produced, water is less and its distribution becomes less, that also affects the process of photosynthesis. Structure of leaf. We have discussed in various adaptations in leaves. Thick cuticle, size of leaves, stomata distribution, all these help the process. So these are internal factors which affect the rate of photosynthesis in plants. Now let's move on to understand two concepts which will help you to understand the experiments which are based on photosynthesis. In the coming video, I'll be discussing with you about the various experiments which are performed to test photosynthesis process. To understand that, we have to clear two concepts: destarching and iodine test. Now, why is destarching required? We know that starch is the output, the end result of photosynthesis process. It is actually glucose, but glucose gets stored in the form of starch. So if starch is tested in leaves, we can test the conduction of photosynthesis process in it. So before we conduct any experiment on photosynthesis, we have to first remove the starch which was already prepared. So we have to do destarching. Destarching means removal of starch. How is it done? It is done by placing a plant in dark for 24 to 48 hours. So when a plant is kept in dark, what happens? Photosynthesis process doesn't get carried out because there is no light. So the stored food is all utilized, and within a day or two, entire stored food starch is removed, it is used up. So thus the plant becomes destarched. After conducting the experiment on photosynthesis, we do iodine test to check that new starch has been produced by the plant or not. So how to do iodine test? There are 4 steps which completes the iodine test. We take a leaf, it is kept in boiling water to kill the cells. Then the same leaf is inserted in methylated spirit. That removes the chlorophyll of the leaf. Then the leaf is put in hot water to soften it. And after that, iodine solution is poured over it and we see a change in color. What change and how? Pictorially, let's understand in this picture. Here you see a leaf is taken and it is being boiled in water. That kills the cell and then it is put in ethanol or methylated spirit. That removes chlorophyll and the leaf becomes, now till it is kept boiled here in ethanol, the leaves become hard and brittle. So it is again placed in hot water to soften it, and after that iodine solution is poured on it. With this, here is a change which is seen. The portion of the leaf which contains starch, that turns blue black, that confirms the presence of starch, and the portion which doesn't have starch, that turns into brown. So with this we conclude that starch was produced and photosynthesis process was performed. With this we complete today's video. Let's do a quick recap. Today we discussed about external and internal factors affecting photosynthesis, destarching process, and iodine test. Please revise the lesson, children, and go through the videos properly to make your concept clear. Thank you and stay safe.